Okay guys, and we're going to finish up with the final biologically important molecule group, the nucleic acids. Remember, I already did the overview video that showed you all three of them together, DNA, RNA, and ATP. So now I'm going to focus on just the DNA molecule. Remember that the monomers for nucleic acids are a nucleotide, which consists of three distinct parts: the nitrogenous base, of which there's 5, adenine, cytosine, guanine, thymine, and uracil. A 5-carbon sugar ring, there's 2 different ones. In DNA, we have deoxyribose. In RNA, we have ribose, and in ATP, we have ribose. And then, of course, the third part of the nucleotide, the phosphate group. So the 3 polymers that you study from the nucleic acids are DNA, RNA, ATP, which I went over in the overview video. So focusing on just DNA by itself, we take a look at the modeling. So here we have the 4 nitrogenous bases: adenine, cytosine, guanine, and thymine. So she might put these out on the lab, practical, as a picture and say, identify the name of this nitrogenous, or what is the name of this model? And this would be a cytosine because it's got a C. That's an adenine. There's a guanine. There's a thymine. And just to show you, there's a U, the uracil, for RNA. And you can see how in these models we've color-coded them differently to show you the difference between DNA with its deoxyribose sugar and RNA with its ribose sugar. And you can tell the difference because if you take a look at the second carbon here, you can see that this one has an oxygen, and here the second carbon doesn't have an oxygen, so that's why it's called deoxyribose. So that's the 4 nucleotides, A, C, G, and T for DNA. And then here is your whole model for DNA. And you can see it there. All right. Double helix structure. All right. Remember, ACGT, you don't need to be able to tell the difference between the nitrogenous bases. There's the deoxyribose sugar, and there's the phosphate group. So that's the DNA molecule, and then there's the 4 bases for DNA.